Hello everyone. In continuation to cell transport mechanism, this is the second part of the lecture. In the second part, we are going to discuss in detail about the active transport. Now, as you know that membrane transport is of active and passive type. So the active transport uh, is of primary and secondary type. The examples of primary active transport are sodium, potassium, and calcium ATPase, whereas the example of secondary active transport is ion driven co-transport. The third mechanism by which active transport is done, it's by vesicles, which is of two types, that is endocytosis and exocytosis. Endocytosis again is of four types, that is receptor mediated endocytosis, phagocytosis, Pinocytosis, and transcytosis. The mechanism by which the active transport works is through uniport and co-transport. Co-transport is again of two types, symport and antiport. The salient features of active transport include that it acts against the concentration gradient, so ATPs or energy is needed, and around 40% of total energy expenditure in a cell is used for active transport system. Active transport is unidirectional. It requires special integral proteins called transporters. These transporters undergo saturation at higher concentration of ions, and they also undergo inhibition by specific compounds which may be of organic or inorganic in nature. Now the types of active transport are primary active transport, which directly uses a source of energy to move molecules across the membrane against their gradient, and the secondary active transport, in which the electrochemical potential difference is created by pumping ions out of the cell, and this is used to transport molecules across the membrane. So these are the two types of active transport. Now this is the most common example of active transport, which is the sodium potassium ion pump. So it is the best example for active transport, where cell has low intracellular sodium, but the concentration of potassium inside the cell is high. As you can see here, high Na and low potassium. This is the extracellular fluid and this is the intracellular fluid, where the sodium is low and potassium is high. This is maintained by the pump known as sodium potassium activated ATPase pump, generally called the sodium pump. It is an integral membrane protein, and it was discovered by Jens Kuh, and he was awarded the Nobel Prize for his work. So as you can see here, the sodium potassium pump brings sodium ions out of the cell. So it is pushing the sodium ions out of the cell, although there's already lot of sodium ions present in this extracellular fluid, and it is pushing the potassium ions inside the cell despite the higher concentration of potassium inside the cell. The hydrolysis of 1 ATP yields expulsion of 3 sodium ions and an influx of 2 potassium ions. The sodium binds to pump, leading to phosphorylation by ATP and release of sodium. Potassium binding removes phosphate and reverts it back, whereas potassium is released. Ion transport and ATP hydrolysis are tightly coupled. Digoxin and obane inhibit this pump. These are the inhibitors of the sodium potassium pump. The second example is the calcium pump, which is an ATP dependent pump which functions to regulate calcium ion concentration, and thereby it regulates muscle contraction. A specialized membrane system called the sarcoplasmic reticulum is found in skeletal muscles, which regulates calcium ion concentration around muscle fibers. In resting muscle, the concentration of calcium around the muscle fiber is low. So stimulation by nerve impulse result in release of calcium. All the calcium is released, triggering a muscle contraction. For each ATP hydrolyzed, 2 calcium ions are transported. So this is the whole neuromuscular uh, contraction mechanism. Now the mechanism, uniport and co-transport. Uniport carries single solute across the membrane. Example, glucose transporter, which is present in most cells. Other examples are calcium pump and also. Co-transport, where transfer of one molecule depends on simultaneous and sequential transfer of another molecule. These are of two types, symport and antiport. In symport, the transporter carries two solutes in the same direction. Example, sodium dependent glucose transport, amino acid transport. And the antiport system carries two solutes in opposite direction, which include sodium pump, chloride bicarbonate exchange in RBC. So this is the complete picture of uniport, where one molecule is being transported. In symport, two molecules in same direction, and in antiport, two molecules in opposite direction are being co-transported across the lipid bilayer membrane. Now you can see that there are three different types of proteins here: the ion channel, ion carrier, and ion pump. The ion channel and ion carrier are used in passive transport, whereas ion pumps are used in active transport, wherein ATP is hydrolyzed and energy is utilized for the transfer of ions. In ion channel and ion carriers, transfer ions from higher ion concentration to lower ion concentration, whereas ion pump transfers ions from lower ion concentration to higher ion concentration. So it works against the concentration gradient, and these works towards the concentration gradient. What about large molecules? So the large molecules are often transported by a vesicular transport system. Now let us try and understand what is the vesicular transport system. So vesicular transport system is of two types, that is endocytosis and exocytosis. Endocytosis again includes phagocytosis, pinocytosis, and receptor mediated endocytosis. So as you can see, in phagocytosis, a solid particle such as bacteria, virus, or any dead cell is engulfed into the membrane, which forms a pseudopodium first, and then this is pinched off as a food vacuole. So this is the endocytosis. Now in pinocytosis is also known as cell drinking, where extracellular fluid is first filled in a membrane vesicle, and this vesicle encloses to form a and pinches off to form a vesicle containing fluid. So this is known as pinocytosis or cell drinking. The third kind of vesicular transport is receptor mediated endocytosis, in which there are here I'll be quoting the example of clathrine coated pits. These are special structures found on where low density lipoprotein is taken in by these clathrine coated pits. These are present in cell membranes. Now when the lipid comes and it attaches to the receptor, this receptor pinches off and it carries the lipoprotein, and this is known as receptor mediated endocytosis because there is a presence of receptor. The last type of vesicular transport is the exocytosis, where the particles are thrown outside the cell or transferred outside the cell. So now what happens is that a vesicle is formed, um, which is full of the molecules or the solid particle which has to be transported outside the cell. It goes and fuses with the plasma membrane, and a small part of it pinches out and the molecules are released outside the cell. So because it is released outside the cell, it is known as exocytosis. Transcytosis is also a type of endocytosis. This is a transport process for macromolecules across the cells, especially epithelial cells. It may be cavially mediated. It involves the entry of pathogens into intestinal mucosal cells and blood-brain barrier. Immunoglobulin A and transferrin and also insulin are some of the molecules transported by with the help of transcytosis. Transcytosis is also an effective mechanism for targeted drug delivery, example antibodies and simple macromolecules. Now let us come to the summary of methods of transport. So uh, we'll see the different transport mechanisms, their types and the material transported. So we'll start with diffusion, which is passive transport. Small molecular weight material is transported. Osmosis, which is again an example of passive transport, and it transports water. Facilitated transport or diffusion is also passive transport, where sodium, potassium, calcium and glucose are transported. Now primary active transport, it is an active form of transport where energy is needed in form of ATP, and sodium, potassium, calcium ions are transported against concentration gradient. In secondary active transport, amino acids and lactose are transported. Phagocytosis is an active transport. It is a vesicular form of transport where large molecules such as whole cells and cellular structures are transported. Pinocytosis and potassium are also examples of active vesicular transport where small molecules or water or liquid is transported. The receptor mediated endocytosis is also an example of active vesicular transport where large quantities of macromolecules are taken inside the cell. In exocytosis, which is also a vesicular form of active transport, where waste materials, proteins for extracellular matrix, and neurotransmitters are exported outside the cell. So, this is the complete summary of different types of methods of transport. Now we come to a specialized type of transport which is known as ATP binding cassette transporters. It is also abbreviated as ABC transporters. These are four different class of membrane bound transport proteins. These include ion channels, transporters, aquaporins, and the last one is ATP powered pumps. So these ATP powered pumps have various kinds, and one of the example is ABC transporter. ABC transporters are uh, present in almost all membranes. These are ubiquitous membrane bound proteins. They can move substrates in flux or efflux of cells. In mammalian cells, ABC transporters are expressed predominantly in liver, intestine, blood-brain barrier, blood testis barrier, placenta and kidney. Now these are various ABC transporters and they transport various substances. For example, ABCA1 transports cholesterol, ABCB4 is a multi-drug resistance transporter, ABCB7 is involved in iron hemostasis, ABCC2 is also implicated in multi-drug resistance associated protein, ABCD1 is involved in anchoring of very long chain fatty acid CoA synthase. ATP7A and ATP7B are copper transporting ATPase. SLC11A2 is known as divalent metal ion transporter, and SLC30A8, it's a defect in receptor associated with impaired pancreatic or beta cell causing defective insulin secretion. So these are various ABC transporters and their functions. So thank you. Hopefully you enjoyed the lecture and enjoyed learning about cell membrane transport. Keep listening and keep watching my videos.